For our next conversation, please join me in welcoming Dr. Nubar Afeyan, founder and CEO of Flagship Pioneering and the co-founder and board chairman of Moderna, and Dr. Ava Carter, a postdoctoral fellow at the Harvard Medical School and a Hannah H. Gray Fellow of the Howard Hughes Medical Institute, who will discuss how the U.S. can continue to push the frontier of biotechnology.
Hello. Thank you all so much for being here. So I'm Ava Carter. I'm Ash's daughter, but I'm also a biomedical researcher. And so it's really a great honor to be hosting this fireside chat with Dr. Nubar Afeyan, who is the founder and CEO of Flagship Pioneering. He's also the co-founder and board chairman of Moderna, the really pioneering messenger RNA medicines company whose COVID vaccine is likely in your arm and is estimated to have saved over 2 million lives. And Dr. Afeyan really rearranged his travel schedule to be here today, so thank you so much. We're really glad to have you.
Great. Good to be here. So maybe I'll just add to the list of presenters this morning that I saw to just say my connection to Ash as well, which may be unusual given that I have not served in the government. Ash joined MIT's board and was actually quite influential over a relatively short period of time. And it happened to be just around the time that I joined as well. So we were equally kind of struggling with the complexity of the task at hand and all the different issues that you heard about this morning. But more importantly, I had a chance there to strike up a discussion with him that I had started with Graham years earlier during the pandemic, having to do with what the world might look like if health became a matter of security as opposed to care, which is an area we can get to. Much of the work that we've done having to do with developing treatments and vaccines and the like leaves one wondering what would happen if instead of waiting for people to be extremely sick and then having really expensive treatments for them, that we actually try to do something the way the defense establishment does, which is to get upstream of the threat and detect it, surveil, delay, deter, derail, all things that I thought this surely would be a good idea. And I thought he's one of the few people that would have had both the world's experiences, both in thinking about policy technology, but also thinking about defense. He immediately pointed out to me some of the small limitations of thinking about defense and health in the same sentence as it relates to the way our government works. So he was quite an educator to me. And unfortunately, our discussions were cut short by his untimely passing. But I'm very pleased to be here as well in his honor to continue the dialogue.
So you began your career in biotechnology really at its emergence nearly 40 years ago at MIT, and a lot has changed since then. So I'm curious if you could just share with us your vision of what the next 40 years look like in this field. What trends and opportunities do you see?
Boy, yeah, that's in 1 minute. Well, I know that you're starting your career. Hopefully, you'll have some involvement as well on the technology side that we were talking before. So yes, I'm an immigrant, as you can probably tell by my name, and I had the great fortune of coming to this country alongside hundreds of thousands of others and millions to go to school. I did a PhD at MIT in the 80s, and unlike what people did at the time of my age or of my background, which was people generally went to either academia or a large company, I ended up starting a company in 1987, which became the first developer of tools for this industry. And I was at the time inspired by a meeting with a person who'd done the same thing 30, 40 years earlier, David Packard, who I had a chance encounter with at a meeting like this. And from there, I decided to be an entrepreneur. And so I've been following a technologist, innovator, entrepreneur role since that time for 38 years. So because I could not tell you where we are today 40 years ago, I will not try to do that today, even though I might have learned a thing or two, because the one thing you realize in this field is that it is, biology and its applications are uniquely a field in which human design is not a precondition for simplifying the problem set. In nature, because of evolution, we're dealing with complexity of a sort that we cannot make the assumptions you can in many other systems, where since you know a human designed it, you know that it's going to be within some limits of how humans operate. Here, that's not an assumption. And so the complexity is extreme, and in that time frame, we've gone from not being able to measure much at all to measuring more and more and finding out just how little we know, not to knowing much, to finding out with the genome sequence just how little we knew. And so I would say being in this field, and this has ramifications on its application, its use, its threats, et cetera, we can come to. But the one thing I've learned over the last 40 years is that I can definitively say that we now know, we know less about disease or biology than we thought we did. And that's been the case every year. So when you get threats like COVID, it's kind of a super humbling moment for humans because we thought we knew about the human immune system. In fact, we knew relatively little of what actually happens, and so on and on. So I think we have to be, this is going to be a very interesting next decade in this space because, and maybe we'll come to that later as well, the advent of generative AI tools and their incorporation in our everyday work, which we do across a dozen or so companies, actually for the first time gives us a tool to deal with the complexity of the science we're studying. The only odd part is that the human is less and less capable of understanding now either system, either the way a generative AI models it or the way nature does it. And so that's a whole nother world that the next 40 years will plague us with.
Yeah, so on that topic of AI, there's obviously huge opportunity in biology with AI. I see it every day. Everyone's applying AI to all the problems that we work on. But there are also dangers of the convergence between biology and AI. For example, using AI to design a pathogen. And so are there particular areas that excite you or worry you in the near term?
Sure. So maybe I'll just step back because most people in the room won't know what it is that we do. So as per the introduction, I'm founder and chairman of Moderna. Moderna being one out of 100 companies that my firm has created using science that we develop in-house. So we operate on the other side of Cambridge, a company that creates companies around science that we develop. The distinct feature of this science is that we're not applying science that's being done in government labs or academia. We're actually creating science that just completely doesn't connect with current reality. And that's what happened in the case of mRNA. There are many fields in which we reach the limit of what we think we can do, and then technological breakthroughs open up the field again, and we do quite a bit of that. The reason I say that is that AI for us over the last 5-6 years has been just such an area. I mean, as you know, in biology, what is called bioinformatics, which was how do you deal with all this complexity in data, was a field that was using machine learning tools, all sorts of machine learning tools, for a very long time before machine learning tools became popular again due to the more recent versions with large language models and transformers. But that field was largely kept alive by among the most applied places being in biology. So for biologists, machine learning and AI isn't a complete revelation, but its use in actually asking questions and creating hypotheses and interpreting text and being able to have read every paper that's been written and apply it to the question in hand, that of course we had not seen before. So let me give maybe a couple of examples of how it is being used, and then we can talk about the potential misuses. So 6 years ago, we started a company called Generate Biomedicines. We had a hypothesis. We asked the question, could you create a sequence of DNA that could essentially deliver any protein function you wanted? So the idea was, could you just create correspondence between DNA sequence and protein function? Now, those of you who remember biology, most of you who probably took biology tried really hard to forget it, but those of you who remember your biology, remember that the central dogma is DNA codes for R, mRNA codes for proteins. Everything we've learned since then is that that dogma occurs once in a while, but there's 100 other ways in which all this happens and controls each other. But one thing is for sure is that DNA is a far cry from a protein function, many, many steps along the way. We thought, could a machine learn how to create direct correspondence between the function we want and DNA? And before you said, like, how can you even say that? And the reason you can, we think, is because when you get your inherited DNA from your parents, there's no instruction manual of how our DNA makes RNA, how RNAs make proteins, how proteins fold. It just works. And so clearly, the function is encoded into the sequence. And we thought, if you gave enough examples of the sequence of a desired function, could the system just come up with more arbitrary sequences? Well, I'll spare you doing your own research because you can go do it. We've actually released our model to do this. But the answer is you can. We trained against thousands of proteins that are bound to other proteins, the sequences, and then said, make me another one for any arbitrary target sequence. And literally a computer can generate 100 sequences. You make 10 of them, and 1 generation later, you have an antibody that's better than any antibody that people develop in the labs. This is not, I'm not making claims, you can look this up. One of the first things we did, speaking of good and bad, is that we said, okay, there's a problem with the spike protein, for example. This was 2 years ago, 3 years ago. Can we generate antibodies to parts of the spike protein that don't mutate? Now we're worried about mutations in the spike, and the virus keeps trying to overcome our immune response. Dirty little secret, our immune response only goes after certain parts of the protein and leaves other parts alone. Why does it leave other parts alone? Because they're non-immunogenic. That's a tautology in definition. In other words, because it doesn't make antibodies, the virus doesn't have to change those because the system, it kind of laughs at the immune system because it can't go after it. A computer doesn't follow what the immune system does. So if you just tell a computer without knowing what's immunogenic or what's not, go after this part, it does, and we did. And we went into the clinic and showed that in humans, you can bind to regions of virus. So that was an interesting application. But now, of course, we've started making literally dozens and dozens of different antibodies to go after any number of targets. Could that technology be used to make proteins that go after positive targets and essentially create a much stronger way to perturb for the negative? Sure. You take that same capability, you apply it to DNA, to RNA. We now increasingly can design with the power of these types of large data-driven models at any levels. Cell level is probably still complex. Organism level is even more. But I think that's one aspect of this. So yes, if you wanted to apply these capabilities to make it even easier to design a virus or components of the virus, you could. And so, but in every other technology, we have the same dilemma, and we have to put safeguards, and I think people are actively working on, I think kind of my view is that regulating AI is not the way to do that, because actually biology itself can do most of these things. In nature, we have a lot of pathogens. The question is, how do we think about using these to protect ourselves, and which we're doing. So a lot more to go, but it's not a wonder that when Jensen Huang was on TV on 60 Minutes about 6 months ago and talked about the most exciting applications of their chips, it was biology in the form of this company and robotics, which you're going to hear about. So I think those are the areas where there's immense opportunity in the complexity, but also some peril if you don't have the right constraints.
So while we're talking about the spike protein, in 2013, Moderna received an award from DARPA to develop novel vaccines using mRNA. I think a lot of people probably think that Moderna came out of the ether right during COVID, but it had really been in development for a long time. And so I'm curious, what did DARPA and other agencies in Moderna see in 2013? How did they see into the future that we should be seeing now? And what role do these public-private partnerships play in pushing us toward this frontier?
Well, DARPA was a special agency in that it always did not focus on adjacent capabilities. It kind of allowed one to take leaps. That's the way we describe it within our own labs. And so in science, just to make this very brief, in science, you're allowed to take extremely careful and well-rationalized steps that are in close proximity to what's already been done or is known. If you went out and said, give me money to do something that's 6 steps removed from where you are, you can't get money for it. Nobody will work on it. DARPA at least creates a bit of a safe space motivated by goals and targets as opposed to capabilities and their adjacencies to allow you to actually do some of that. And so they were looking, for example, at the time among their many projects, for ways in the battlefield to be able to detect the threat and make a neutralizing antibody to it. And that's how we first started interacting with them. It was not in the vaccine space. It was in the antibody space. So could you sequence in the field a threat and then immediately turn around and make an antibody very rapidly, and then it expanded to can you make a vaccine very rapidly? So how did they detect it? Well, they detect everything. So that's their job, is to survey it. And this was certainly something that was not yet known not to work. I always teach when I used to teach at MIT and in our business school in innovation. I used to tell people that when you're in the innovation and startup world, you have to learn that everything you're working on is simply not yet known not to work. And once in a while, it actually fails that test and it does work, as opposed to thinking everything you're working on is going to work. So RNA clearly at the time in the hands of Moderna had already shown, at least not publicly, but we had shown them privately, that it has this very rapid turnaround time. You can think about design of mRNA. And this was the early days. But the one thing I'll tell you, since everybody lived through the COVID period, The general public didn't know about mRNA as a therapeutic. That's true. They also didn't know about the company Moderna as a result. Moderna is in Kendall Square. But in 2020, the company had 700 people and had spent $2.5 billion developing an mRNA platform, had taken 9 vaccines into human clinical trials. In all 9 cases, we had neutralized the virus. Vaccines are very unusual in that in phase 1 trials, you pretty much know if you can get an immune response or not. Unlike cancer, where you have to wait till dose ranging and this and that, it's a very quick readout. You still have to show safety. And so what was this overnight phenomenon that everybody's worried has never been tried before, et cetera? It was an interesting question. I heard a lot of discussion in the morning about risk, and we think of risk and uncertainty as very different things. How risky was it that we were going to be able to take RNA and make a vaccine for a 10th virus, namely this one? And because we had collaborated with DARPA, because as a result we had then further collaborated with BARDA, because also in parallel we'd worked with NIH, long before COVID showed up, in NIH is Tony Fauci's group. So 2 years before this, we had partnered with Tony Fauci's group to apply this mRNA platform to the MERS cousin of SARS-CoV-2, and we were working towards that program. Why I say that is that the underlying platform was actually ready but untested based on its speed. And so that was but I do want to take opportunity because of the format of, or the context of the discussion to say that beyond DARPA, which actually Ash and I had a long discussion about kind of how DARPA's kind of involvement helped us create credibility to the field. That was an interesting side benefit, is that as they started increasingly funding this activity, even though it was 1/100th of the funding that we had obtained through the private market, it was highly symbolic because their selectivity, their involvement meant a lot for us to be able to show the outside world that this is, because nobody had ever seen anything like this before, and therefore in science, as you know, we're trained not to believe anything that's new until there's a lot of testing. But the thing about DARPA, BARDA, NIH is even beyond, is that when the pandemic hit, and I'm saying this, as we come up to an election, which is kind of quite ironic, is that the single, in my view, biggest externality that caused all this to happen is the creation of Operation Warp Speed. It's unfortunate that that whole experience was politicized, then suppressed, then with the change of administration became a distant memory, and then that experience, which I think was about as exemplary a collaboration between private and public sector in a non-wartime, that felt like a wartime to many, was able to massively change the course of what we were able to do in ways that newspapers covered of saying, oh, the government's giving all this money, how come they're giving all this money? What people didn't really understand is behind the scenes that the War Powers Act was enacted. Every supplier that we were getting anything from essentially had to supply us before anybody else got their supplies from all over the world. The fact that the military supply chain through General Perna was brought to bear to help us do things in timeframes that were weeks and months shorter than it would have otherwise happened. Of course, Ash knew of all this. And then the clinical trials network. I mean, the notion, let me just say one thing, not to kind of sensationalize it, but just for you to understand the moment. So yes, we had developed a platform. Yes, we had tested in a lot of humans. We had ever made 1,000 batches of anything, 1,000 vials of anything. If this was to work, we were going to have to make hundreds of millions. So the question as an engineer, which I was trained in as undergraduate, is how do you go from 1,000 doses to 100 million or 1 billion in 6 months, right? So that is without any precedent, because unlike in spaces where you just make big reactors and you just make larger quantities, this is a fairly miniature technology, because you're literally designing something and putting it into particles, and every dose is a few milligrams. So that the whole process of scaling up and then testing in parallel in 30,000 people in a span of a month or two could not have happened without that level of organization and determination. And most importantly, from an economic signaling standpoint, the government said, anybody, not just our technology, anybody who can produce an effective, safe vaccine, they would have 100 million or 300 million doses purchased at this price. And what that did is it allowed the private sector, like our company and others, to now begin to spend risk capital, knowing that there was certainty of what the price and demand would be. Yeah. Without those things, we would have had a vaccine in 2 or 3 years. No doubt about it, in my mind. And I'm saying that as being deeply involved in it. It's really, and I remember Graham at the time, and I used to say we should really kind of make this an example and this and that, but it was like long buried before we could do anything positive about it. But I do think that if we think about climate-driven health threats that will come next most assuredly, that if we think about what to do with carbon, we could take a lesson from what happened with OWS, and if we seriously as a society want an outcome, we should take the DARPA model that kind of produces proof of concept and actually create the incentives, the clarification of the regulatory process, not the elimination, but the clarification, sufficient to get the job done. And I have no doubt that there's a lot more capacity in solving what seem like unimaginably complex things, very much based on what we already have, if only we could get this organization put in place. And unfortunately, we're very far from that.
Well, it's really enlightening to hear sort of the origin story of how that happened. I'm curious, you talked about the complexity of biological systems, right? Evolution doesn't make them the way we would engineer them. And so they're really complex, and then the risks in testing new therapies can be really quite huge. Human lives are at stake. And so this can make the pace of innovation really slow, not just in terms of incentives, but actually the pace of the research. And so how do you evaluate companies when you know that, or how do you evaluate projects when you know that the time span is going to be very long? And how do you look that far into the future and balance that sort of risk of failure rates?
Well, so let me explain kind of how we think a bit differently, and that gives us a bit of an advantage, albeit I'm sure we have many other disadvantages. The way we heard this morning from the heads of R&D, the establishment at Harvard, MIT, this notion that you do basic research and that research might have applications, then there's some additional people who come in and do translation, and then that translation sometimes has commercial application. And hidden in that is a sequentialism, albeit unpredictable in which ones are going to advance in what pace, that is the way, so we do innovation following the arrow of time, so present, towards the future. We've tried to figure out how do we create scientific advances today that are in service of what we would like to create and what we would like to see exist. And it turns out that if you prosecute present-day science from the future backwards, so that you actually have a desired capability that you want to enable, you don't have to work on every which direction of science to be able to do that. You can kind of reach backwards and say, still you have scientific questions. It's not an application anymore. It's what we call reverse translation. In other words, pick a destination of a capability that you feel creates a major solution and come backwards and say, what do I have to believe to be able to get there from here? It seems like an obvious idea, but I can tell you there's no funding for that. There's no patience for that. And no venture capitalist, no peer review panel at NIH, no company will ever fund that. So why I'm saying that is that the way we do it, we already know that the thing has significance and can create value before we ask the scientific questions. And so it's the science is in service of not just a proximal application. It could be a completely. I mean, I'll give you an example just to get people to think through. We do this by asking these what-if questions. We ask hundreds of what-if questions a year. One of the questions we asked is, what if there are proteins being made in our bodies that we don't know exist? You can't ask that question to the scientists. They'll laugh you out of any conference because you're only allowed to ask questions about the proteins that are known to exist, or maybe one or two that you missed. We were asking, what if we're wrong, completely wrong about this? And if you said, why would you ask this question? You go back 20 years and we were wrong about much of our assumptions about the human genome. That's just 20 years ago, 35 years ago. We thought we had 100,000 genes. Turns out we had 20,000. We thought, every gene makes one protein. Now we found that. So we asked this question 4 or 5 years ago. What if there are other ways that the body makes proteins? This would matter because that means everything we're studying about cancer, there's a whole chapter missing. Everything we're studying about the immune system, everything we're studying about neurons. Okay, so if you ask that question, then you come backwards and you say, okay, assume there are such proteins, where would they be? And the world looks completely different. So we asked this question, we start coming backwards, we said, okay, well, you should see these in mass spectrometry readouts that we all make. Turns out there's tens of thousands of mass spectrometry readouts that have no explanation in the genome. And then people consider that noise. The scientists are very good at thinking everything they don't understand is noise, and then everything. Then we can, it's true as a scientist in the rooms, then you come backwards and say, well, you should see these proteins somehow being translated by ribosomes. So we just look at as you know well in your lab work, RiboSeq is a technique that's relatively new. We looked at that, and there's tens of thousands of pieces of RNA that are being translated for which we have no explanation. And put all the pieces together, we now have, and NIH has now begun funding several years later, a lot of work in this area, now very new work. We now have the belief that there's tens of thousands of what's called open reading frames from the non-coding part of the human genome that are expressed in proteins. They're involved in cancer, we have direct associations. So when you said, how do you know what will work, what won't work? I think when you think about platforms, you heard from Eric Schmidt this morning, the idea of platforms in tech. I think we need to think about platforms in biology, in that we need to think about common advances that can generate many, many products. And the patience has to be that you might not know which is the most obvious product to get out of a platform, but a platform enables a lot of different, what they call shots on goal in this business. And from there, you got to be able to survive long enough to have one of them go in. That's kind of how we think about not sitting there and predicting based on the beauty of the paper or the intelligence of the inventor, what's the chance it's going to succeed? That's the investor mindset. We instead just run the evolutionary experiment.
Wow. So in looking for those ideas and people that think that way, you spend a lot of your time traveling and engaging overseas. And so where do you see opportunities internationally for advances in biotechnology, either from scientists over there or recruiting talent?
Well, of course, yeah, I mean, there's international and then there's China these days. And so obviously there's a lot that's going on in China and has been for quite a while, both in the supply chain side of this industry, which was somewhat exposed, kind of during COVID and thereafter as everything shut down. And so I think there's a lot of concern and consternation about how stable are we and how self-sufficient are we in being able to make anything we want. That's one side. The other side is just labs and innovation, where they've clearly, one of their major vectors in their strategic plan has been to become leaders in the space of biotechnology, both the capability side, the information side. And of course, there are facilities they have in terms of societal advantages in that the privacy laws that apply here are very different there. You can get access to a massive amount of genetic information, on and on, let alone AI and how it will advance these things. So that's a big kind of piece of the story. The other piece is Europe, and I just came back at 5 o'clock this morning from the Gulf, from Abu Dhabi. There's a huge amount of interest and investment now that they're trying to make in this area. One thing pandemic taught everybody is that they needed national resilience against these kinds of threats. Generally, the US, Europe did not do a good job to cause anybody in the rest of the world to think that they were protected, because we were going to protect ourselves. And the fact that vaccines did not make it to a lot of places which needed it perhaps more in certain cases in terms of the impact, but had to wait till there was massive vaccinations in these Western countries, it created massive stress for us as a company, because we were aspiring to be a global company, and we were, of course, reduced to a pretty local company in that regard. So I think some lessons have been learned. Part of it is everybody wants their own plants, which is not the answer, because everybody shouldn't be making their own iPhones just because they can't get access to iPhones during a war. That mindset is now set in public health. We're trying to work with people to say, okay, how can we make sure that next time around the capability exists, it's diversified, it's available around the world, and you're not facing the situation where everything is shut down kind of due to emergency conditions. So that's getting a lot of attention. I will say again that I think the future, because we're going to run out of time soon, the future, I hope, involves a lot more money, government money, not only here, but the UK and others are now taking this up, working upstream of disease. If we're looking at our 20% of our GDP going into healthcare, that's not going to be any less than 25 or 30 over the next decade or two, unless we can take steps to move upstream of disease. We think of disease as something you get when your doctor tells you you have it. We have a fatalistic view of disease, unless we're doctors, in which case we know that we could have said you had the disease 5 years earlier, but we don't have anything to do about it, so we don't tell you, so we don't worry you. I'm sorry to say, that's the reality. So in general, I'm not a doctor, but I've worked with them for 38 years closely. In general, healthcare, dirty little secret, is a euphemism for sick care. The way you know that is that you only get any of it when you're sick. And yet, if our physical security was only protected only when we were massively attacked, we wouldn't be a happy tribe. right? So we demand our government take care of our physical security long before we're attacked. Well, in healthcare, we go, you know what? If you're attacked and most of you are kind of in deep trouble, we'll come and save you. That mindset had to have been in the early days of the War Department and not the Defense Department, a very expensive proposition. And then people said, you know what? I know how to make this somewhat more affordable. Let's move upstream. In the health world, that seems impossible. Not scientifically. It used to be impossible scientifically because we didn't understand how diseases happen. We now can tell you, we have several companies of ours because we're insisting on doing this, if only for a societal good. We can tell you 6 months, 9 months, 12 months before you're going to get a specific diagnosis based on data at the population level, data at the disease level, and molecular data. This is not markers for late-stage disease, this is markers for pre-disease. In obesity, we can do that today. In type 2 diabetes, we do that today. We know that things like statins help prevent long-term if they're used at low doses. And then we know we can do this in colon cancer. You see a polyp, you take it out. A polyp's not a cancer. Very often it might become one. So imagine a world in which we practice preemptive care on pre-disease humans, not patients, in order to pre-treat what they're going to have. To do that, you need a coordination at a government level because the regulatory agency has to have that as a goal. Society has to have that as a goal. The insurance system won't pay for that today because everybody says, oh, you're going to change insurance every 3 years. Why would I pay for your improved, let alone gyms? I mean, they don't even want to, they will not. So I say all that because I do think that some version of the thinking that is coming out of the protection of the nation has to apply to our health. Whether it's natural threats, of which nature is doing a pretty good job, a lot better job than terrorists are in generating threats towards us, but also man-made threats, which I can assure you the level of that risk is way higher than it was before the pandemic because you have more technology, but you also have more knowledge about the vulnerabilities of the immune system, the ones that are giving us long COVID, the ones that are giving us all sorts of new challenges. Now we not know much better. So those are vulnerabilities.
Yeah. So we have a couple of minutes left, and it sounds like you want to turn healthcare on its head, you want to turn research on its head, and you'll need new people to do that. And so I'm curious, I heard your commencement speech at MIT last year, and I'm wondering if you can speak to the students and trainees in this room about sort of how to tackle those impossible missions and how to start down this road.
Well, yeah, thanks. So if any of you have 12 minutes to spare some time, feel free to go and see a kind of a crazy address I gave. It was in the middle of encampments on MIT's campus. It was a pretty rough period as a result of the moment we live in. And yet there's these students, these armies of people that are coming out with tools and capabilities and aspirations to impact the world. And one of the things maybe I'll leave you with is what I was talking about there, which is that if you're given the gift of an education and you're exposed to mindsets that you really should go after meaningful problems, then you really are signed up to kind of a Mission Impossible team. Right? It also happens to rhyme with the MIT or at least match the MIT letters. And I said, if you realize that the only things worth working on are what seem like impossible missions, then you worry less about whether you're going to be able to create value if you pick these missions correctly. But the one thing that you have to be used to is the discomfort that comes with that, the apprehension that comes with everybody not telling you that it's likely going to work or that you should. Or everything is turned upside down when you don't work on adjacent problems. And so how do you get people prepared for that? One of the great advantages we have, again, we heard a lot about this today, is that what generative AI and broadly AI tools are going to be able to do, I believe will be a unprecedented prosthesis for the human imagination. And if you're going to work on impossible things, you need to exercise your imagination. You cannot derive things that nobody can imagine can happen, note the word imagine, and therefore you won't work on it. You can talk yourself out of it. And I've had a little bit of interaction in the last couple of years with the intelligence community, and I've tried to learn how they think about what we confront all the time, which is anytime you work in science, the distant future is uncertain. We are trained as humans to put everything in the context of risk. So if you ask me in 2010 when we started working on mRNA, what was the probability of success that mRNA would be a drug? I can assure you when we did ask lots of people, people would say, extremely low, or you should use a 50% discount rate per year if you're going to invest, etc. That's a human tendency where we want to say everything has risk. But actually, that was squarely in the category of uncertainty, which is unknown, unestimable probabilities. And so how can we underwrite working on uncertainty long enough to resolve the uncertainty and take an idea into the realm of now risk, but based on data we generate, so that we can now look at it based on data and say, okay, this looks like that, and I think the probability of success is 30%, 10%, and off we go. So the idea of a Mission Impossible was, are you willing to reach, and why should you reach into spaces that are considered uncertain? And there's one simple answer to that. I emphatically believe that high value, relatively low risk opportunities exist in uncertainty. They do not exist in certain risk-oriented places because so much of the competition is there, they will have already taken it away. So interestingly, if you said, are tomorrow's solutions available today? I would argue by definition they are, because otherwise we wouldn't have them tomorrow. And so if I assert that something is going to be possible, then people who are willing to work on it without the comfort of it looking like it's likely going to work may have a portal to lower risk, not necessarily guaranteed, but what's the chance that something has never been imagined or done before is going to be low probability success? Well, it turns out eventually, let me give one last example as we wrap up. In the Moderna case, we worked on cancer drugs, we worked on rare diseases, we worked on many, many things, including vaccines. We had no idea vaccines would be the absolute kind of easiest thing to do. Why? Because you give 1 or 2 doses, it's a specific sequence, and the immune system does the rest. So it turned out in hindsight that it was an ideal thing to make a genetic medicine. But because we were the first to underwrite the uncertainty, we were the first to do the experiments, we found out that in the distribution of risks hidden in that mRNA space, vaccines were the lowest risk. And therefore, we can actually put our resources disproportionately in that. I think that that's a way to think about, that we heard this morning about the science kind of endless frontier. If we were going to write that again, we should think about working on uncertainty in a way that today there's no grant-based way to work on, generally, because no peer will say something that's unestimable as to its risk should be funded. No peer review does that. And so anything peer review throws out because they think it's too risky, I think should go into a separate category, and we should try to figure out, is there an experiment or two that will move it back into the risk realm? Yeah. And those ones, I believe, will be the breakthroughs of tomorrow.
Well, thank you. We're out of time, but I want to thank you so much for being here and for all of you for listening.
Thank you.
Thank you.